7th.
Okay. Anyhow, I welcome you for this today's BAQ 3 online academic counseling session of diploma in aquaculture. I think today is the beginning of this course counseling sessions. So far, whatever the BAQ 1, 2 sessions we have already uploaded in our RC Vishakhapatnam YouTube channel. The missed students can watch that one and they can prepare for their examination as well as the assignment preparation. They can use it. Now I request academic counselor Mr. Loveras, please take the session.
Okay, sir. Thank you, sir.
Yeah, thank you.
So, good evening, everyone. So, today class, today discuss about the coastal aquaculture. So, in previous basic principles of aquaculture, like what is freshwater and what is brackish water and what is marine water. And now discuss about coastal aquaculture. So, in this paper, first of all, discuss about hatchery technology for prawns and fishes. So hatchery technology for prawn and shrimp. So in this hatchery, the main aim is production of seed, like the seed production. So nowadays we have many of hatcheries. In that hatcheries to produce millions of seed, especially either fish seed or shrimp seed. So, like the hatchery aim of hatchery is to produce stockable size of fish or shrimp from the eggs. So, production of hatchery seed and larval technology of both shrimp and fishes are discussed in different larval stages, like especially in this hatchery mean first of all properly maintain of brood stock management. So especially either male brooders and female brooders. So after development or maturation of both male and female brooders, so and after releasing millions of eggs, the eggs are hatched and produced millions of seed, either fish seed or shrimp seed. So the so in this hatchery production, okay. So in this hatchery having some different types of larval stages. So first of all, any shellfishes, especially either prawn or shrimp, having some different larval stages. The first one is Naplia, Mysis, Joya. So in this session to discuss Naplia larval stage. So Naplia is the first stage of larva, either prawn or shrimp. So the egg of peanut prawn hatched into Naplius. So the egg of peanut prawns hatches into Naplius. The Naplius is pear-shaped with three pairs of appendages. So especially three pairs of appendages, it does not have any functional digestive tract or mouth parts. They swim intermittently with their antenna and mandibules. And also, there are positively phototactic characters. There are 6 substages in the Naples larva with a different number of spines at each side of the uh, stage. So each stage lasts for a period of 4 to 8 hours. 4 to 8 hours. So each in this synapse stage having total 6 sublateral stages. So each stage having 4 to 8 hours. So after completion of the total 6 larval stages, so to produce larval stage zoea. So the zoea body of the body of zoea is more elongated than the naples. So the body consists of the carapace. The body consists of carapace, thorax, and abdomen. They swim continuously through antennal propulsion. They have functional digestive tract and filter feeders. They feed mainly on unicellular algal present in the medium. So there are 3 substages in Zoea, especially how many substages are present in Naplia stage? So like 6 substages are present in Naplia. And what about substages present in Zoea? So the Zoea substages are 3 substages like Zoea 1, Zoea 2 and Zoea 3. That means substage 1, Zoea substage 2, Zoea substage 3. So the Zoea 1 is characterized by the development of sessile eyes in the carapace, while in Zoea 2 is eye stalk development. In Zoea 3, there is a development of dorso median spines in the abdomen. So each substage lasts about 24 to 48 hours depending on the temperature of water and feed availability. Like and third one is coming. The next one is the larval stage mysis. So mysis swim through the flexing their abdomen with the head pointing downwards. So there are 3 substages in the mysis. So how many substages in the larva mysis? So the mysis contain 3 sublarval stages. So that is M1, M2 and M3. So M1, that means mysis 1 is characterized by the appearance of primordial pleopod buds and hepatic spine in the carapace. In the mysis 2 stage, elongated and unsegmented pleopods are present, while in M3, that means mysis 1 or 3 is the pleopods become segmented. So each substage was for 24 to 36 hours, and mysis generally feeds on phytoplankton as well as zooplankton. So, and after completion of mysis stage, so the larva is called as PL stage. That means PL means post larva. So the post larva resembles the adult shrimp, and it can be distinguished from the mysis stage by the presence of sepae on the pleopods, which are functional. The post larva also prefer to cling to the solid surface and the walls of the tank. Substages of post larva are called as PL1, PL2, PL3, and up to PL12. So wherein the number of indicates the number of days of post larvae. So post larvae feeds on zooplankton and other dead organic matter. So they can tolerate different salinity ranges. So in this session, some discuss about the uh, identification features of larval stages of Penis monodon. So for example, the hatching Penis monodon, the total lifespan is 12 to 15 hours after spanning. So the identification features is uh, perical spines 1 plus 1. The setae on appendages are not pulmos. And the first is larva Naplia 1. So Naplia 1 uh, after Naplia 2. So in this lifespan also 5 to 6 hours. So characters are perical spines are 1 plus 1. The setae on appendages are pulmos. That's mean well developed. So and Naples 3. So same as lifespan is 5 to 6 hours. And identification features are uh, perical spines 3 plus 3. And after Naples 4, so lifespan is 5 to 6 hours. So identification characters are spherical spines are 4 plus 4. And after Naples 5, so lifespan is 5 to 6 hours and spherical spines are 5 plus 5. And finally, the Naples stage, sixth Naples stage. So the lifespan is 15 to 20 hours. So the identification characters are Percal spines 6 plus 6, then 8 plus 7. And after coming to the next one is Joya 1. So Joya substage 1. That's mean the lifespan is 1.5 day to 2 days. So the identification characters is no eye stalls. And coming to the next one, Joya 2. So the lifespan is 1 day. So identifying characters is stalked eyes. And after Joya 3, so lifespan is 1.5 day to 2 days. So identifying characters is a pair of biromus uh, uropods and dorsomedian spines on abdomen. And coming to the next one is mysis. So first stage like M1. So lifespan is 1.5 day to 2 days. So identifying character is rudiments of leopards like mysis swim with its head down. So and after M2, that's mean mysis substrate 2. So lifespan is 1 day. So identifying characters is unsegmented pleopods with a terminal setae. Unsegmented pleopods with a terminal setae. And after coming to the next one is mysis 3. So lifespan is 1.5 day to 2 days. So identification features is segmented pleopods with terminal setae. And after TL1, so that is lifespan is 1 day. So identification characters is segmented pleopods with setae all over like post larvae sim horizontally. So these are the main uh, either identification characters. Next. So coming to the next one is induced breeding technology. So this is also called as hypopagation. So breeders can be obtained either from wild or from broodstock development in captivity. So breeders obtained from wild normally will not respond to hormonal manipulation unless otherwise it is acclimatized for few days. The problem faced in induced breeding is that variations in gonadal development among individual fish occurs in natural populations as well as in broodstock maintained in captivity. So successful induced breeding depends upon the selection of the recipient fish at the proper stage of the gonads development. Earlier, external characteristics like fullness of belly, color, color and state of swelling of cloaca, such as protruding pinkish and reddish genital papilla, and softness and resilience of the belly, so in female especially, so roughness of pectoral fins, presence of head tubercles in especially males. So we are considered for the selection of breeders. So the more reliable method to access the maturity of females now being used to take a sample of the ova using a catcher and to examine under the microscope. So the diameter of the ova are also observed. The matured ova will have round shape. In case of the males in mature specimens, milk oozes out in genital pressure on the abdomen. And hormones and administration. So hormones have profound effect on induced breeding of fishes. So you will study about the various hormones and techniques to administer them for induced breeding in the following sections. Like the first one is the pituitary extraction. So pituitary gland collected from maturing and mature fishes are used for induced breeding of fishes. So pituitary glands collected from maturing and mature fishes are used for induced breeding of fishes. This technique is called also hypopagation. Is nothing but induced breeding. Induced breeding. So the technique involves collection of pituitary glands, preservation in 100% of alcohol, preparation of pituitary extract, and administration by intermuscular injection. So freshly collected glands are also used. Pituitary glands are weighted and homogenized in a tissue homogenizer with distilled or saline water. The homogenate is centrifuged and the supernatant clear solution is injected to the females and males. So normally the pituitary extract is administered to females in 2 doses and 6 hours. In 2 doses at 6 hours intervals and for males in single dose. The dose will vary from species to species and depending on the stage of maturity and determined by ova diameter measurements. And generally a dose of 3 to 5 milligrams per kg for first dose and 5 to 8 milligrams per kg for the second dose for females and a single dose of 3 to 5 milligrams per kg for males were found to be effective in case of calves. This has been successfully practiced in the breeding of Indian and exotic calves. When glands collected from the same species is used, it is called as homoplastic, and when gland is obtained from a different species, it is also called as heteroplastic. So That's mean homoplastic and heteroplastic. Homoplastic mean so the donor and a receptor both are in same species. That is called as homoplastic type of method. Or donor and receptor both are different species. That is called as heteroplastic type of method. So pituitary glands have been successfully used in the breeding of brackish water fishes like mullet, cephalus also. So in this injection like ovaprene. So actually nowadays having some different types of synthetic hormones like ovaprene, ovapel or ovatide. So many organizations are used in the three varieties of synthetic hormones. So in this session, discuss about Vavaprim. So Vavaprim is a new product now available in the market, which is extensively being used in the induced breeding of freshwater Indian and exotic carps. So it is in a solution. It is in a solution from can be directly used. In majority of cases, the fish breeds with a single dose of 0.5 milligrams per kg body weight. So, and presently, Ovaprem has been found effective in the breeding of brackish water fishes like a Lattice calcarifer and Miguel cephalus and Miguel cephalus and Lattice calcarifer. In Miguel cephalus, it is more effectively when used along with human chorionic gonadotropin. So that is HCG, human chorionic gonadotropin, at an interval of 24 hours between the first and second dose. The initial dose is called as priming dose, whereas the second dose is called as resolving dose. The first dose is called as priming dose, whereas the second dose is called as resolving dose. So, and what about HCG? So, HCG mean human chorionic gonadotropin is another hormone available in the market and commonly used in induced breeding of fishes. It is powder formed in ampoules. HCG is mainly used as a priming dose and pituitary extract. Vavaprem or luteinizing hormone releasing hormone analog are used a resolving dose. For administration of HCG, uh, it is dissolved in distilled water or saline water and the required quantity is drawn to a syringe and administered. The dose of HCG varies from species to species and depending on the maturity stage. And coming to the next one is luteinizing hormone releasing hormone analog LHRHA. So luteinizing hormone releasing hormone analog is an improved hormone available for the breeding of fishes. So due to its high cost, it is not generally used, not generally used, especially in freshwater fish breeding. So luteinizing hormone releasing hormone analog is a normally used and resolving dose where HCG or pituitary extract is used as a priming dose. So in breeding of sea bass like Lattice calcarifer, a single dose at 50 to 100 micrograms per 1 kg body weight has been found very effective. So, in case of the stripped mullet is used in a resolving dose like at 2 100 micrograms per 1 kg body weight after an interval of 24 hours of the priming dose of SCG. Next, induced maturation. So in penis monodon, males weighing above 40 grams are found to be mature in estuary water. So while females do not mature in captivity or brackish water, in nature females move to the sea for attaining maturity. The female maturity is governed by a set of neurohormones secreted by X organ situated in the ostalt and thoracic ganglion. So the gonad inhibiting hormone is secreted by the X organ and the gonad stimulating hormone is secreted by the thoracic ganglion. The gonadic GIH, that's mean gonad inhibiting hormone, is reported to be more active in uterine conditions and in captivity, thereby preventing maturation. So removal of one eye stalk helps to reduce gonad inhibiting hormone level and accelerate ovaprime development. And come to the next one is selection of females. So how to select female species, like healthy hard-celled females, healthy and hard-celled females weighing about 90 grams, having spermatophores in the thelicum with all appendages should be selected from the adults in the holding tanks for induction of maturity. So newly molted or ready to molt animals should be avoided. And sex identification. So how to find out this sex identification in prawns? Like identification of male and female species. So sex can be identified by the looking at the ventral side of the prawn. Ventral side of the prawn. So male prawn can be distinguished by the presence of semi-tubular structure, so known as patasma, located in between the first pair of swimming legs. The female tiger prawn possesses an oval-shaped structure that is called as thelicum, situated in the space between the three pairs of the walking legs. Like ovarian maturation stages. Like first stage one, like, that is called immature stage. Like ovary is then translucent, like immature characters of a female organism. Like ovary is thin, transparent, not visible to the naked eye through the exoskeleton. And stage two, that's mean early maturing stage. Ovary is thin, increase in size in the anterior and middle lobes, visible as thin linear band through the exoskeleton. And stage 3, that is late maturing. Ovary is visible to the naked eye through the exoskeleton as a thick dark linear band expanded at the first abdominal segment. Expanded at the first abdominal segment. And after stage 4, like mature stage, ovary is dark green in color, assume a distinct space, clearly visible at the first abdominal segment. Clearly visible at the first abdominal segment. On stage 5, that is called spent stage. So ovaries appear as a thin linear band, as in the case of stage 1, partially spent ovaries having either the anterior or posterior lobes. So these are the different maturation stages of ovarian maturation stages of prawn or shrimp. And after coming to the next one is eye stalk ablation. So unilateral eye stalk ablation should be done either by cutting the eye or electroculation should be done as quickly as possible. Of the two methods, electrocalculating as an advantage since it is minimum stress, like stocking. So the able prawns are stocked in the maturation tanks with unable males at 4 numbers per meter square with a water depth of 80 to 100 centimeters. So stocking of females and males in the ratio of 2 females and 1 male is desirable. And after seawater quality, it is essential to maintain good water quality in maturation tank. The seawater obtained either from the sea or from borewell fixed in the intertidal zone is treated with calcium hypochloride, vaguely aerated to expel chlorine and allowed to settle for 6 to 12 hours. So the residual chlorine is neutralized with sodium thiosulfate and the water is then passed through the sand filter. EDTA is added at 10 ppm as a chelating agent. The treated seawater is used for passing through 10 micron fabric filter for broodstock and maturation and 5 microfilters for spanning and hatching. And after coming to the next one is feeds. That's mean feeding of brooders. It is essential to feed able shrimps with quality feeds to maintain spanners with quality eggs. And after fresh feeds like clam, squid, mussel, oyster, crab meal, so having similar amino acid profile of prawn, which are rich in long chain polyunsaturated fatty acids, are used as maturation diet. Pellet feed with 50 percentage of protein and 10 percentage of pupa, that's mean polyunsaturated fatty acids, that is also called as pupa, are to be provided for the achieving good result. And after reaching hatchery, the prawn should be gradually acclimatized to seawater salinity. The prawns should be disinfected in 25 to 50 ppm formalin for 1 hour before transferring them into the broodstock holding tanks. Broodstock can be maintained in holding tanks for a period of 1 month, changing 100% of seawater of 34 to 36 ppt so daily and using fresh and pellet feed. And coming to the next one is optimum conditions for shrimp maturation in captivity. Optimum conditions for shrimp maturation in captivity. The seawater quality and photo period to be maintained for maturation of shrimp, especially Phineas monodon in captivity. So first of all, based on this, some different parameters like first one is tank size. So the tank size is 5 to 10 tons capacity. So circular or rectangular tanks made up of fiber reinforced plastic or concrete tanks. And second parameter is light. So light is reduced or dim light. And third one is light quality. So light quality is either blue or green color. Photo period. So photo period is 12 hours light and 12 hours dark. That's mean 1:1 ratio. Like 12 hours light and after 12 hours dark. And water depth. So water depth is 8 to that's mean 80 centimeter to 100 centimeters. And another one is salinity. Salinity is should be maintained 30 to 36 ppt. ppt means parts per thousand. And after temperature, so temperature is 28 degrees of centigrade to 60 degrees of centigrade. So not only this is the optimum temperature, especially the person should be maintained 28 to 30 degrees of centigrade of temperature. And pH is 8 to 8.5. So dissolved oxygen is uh, saturation by continuous aeration. So stocking rate is 4 numbers per meter square. 4 numbers is sufficient per meter square of place. And after stocking size, so females are 90 to 150 grams and males are 60 to 80 grams. Sex ratio, two females and two females and one male is that is the sex ratio of shrimp maturation captivity. And water management, 100% exchange or day using filters or 200% exchange by flow through system per day. And after feeds like clams, mussels, squid, crab, oyster meat, and 15% of the 15% of the total weight per day. So polychaete worms at 6% or Artemia biomass or at 2.3% of the total biomass. And pellet, that's mean artificial feed. So 2% of the total biomass per day. And feeding schedule 4 times in a day. 4 times in a day. That's mean 24 hours. And coming to the next one is spanning and hatching. Spanning and hatching. The progress of ovarian development in the ablated prawn is monitored 3 days after eye ablation by using an underwater light. The ablated prawn start maturing few days after eye ablation. The mature prawn in stage 4 should be transferred to the spanning tank after disinfecting with 50 ppm formalin for 30 to 60 minutes. The mature prawn are transferred to the spanning tanks. So at one prawn per tank filled with filtered seawater and provided gentle aeration. Spanning usually takes place during early morning hours as indicated by foam brown or light pink in color attached to the sides of the tank. So the mature prawn are returned to the maturation tank. The eggs are siphoned out, cleaned with seawater and transferred to 4 tons 4 ton capacity 4 tons capacity like either tank or basin. After through mixing, two 10 ml samples are taken and the number of eggs are counted which are raised to the total volume of the water to obtain the total number of eggs released by the spanner. So evaluation of quality of eggs is done by observing 100 eggs under a microscope. The eggs from each spanning are transferred to hatching tanks so containing filtered seawater and provided gentle aeration. The eggs having normal development normal development hatch out into nuclei in 15 to 17 hours after spanning, depending on the temperature. So able female weighing 90 to 150 grams span produce like 1 lakh to 5 lakhs eggs, 1 lakh to 5 lakhs eggs with an average of 2 lakhs eggs per prawn. So the quality of eggs decrease with repeated spanning. So that is the spanning and hatching capacity of Penis monodon. And coming to the next one is broodstock development and breeding of fishes. Broodstock development and breeding of fishes. For an overall and integrated backwater aquaculture development, finfish culture should also form an integral part. The monospecies farming practices was the main reason for the outbreak of diseases in the recent past, or many shrimp farms were closed down. India is best world with a esteemed 0.9 million hectare of the brackish water area. The water bodies can best be utilized for culturing shellfishes and finfishes. Breeding and seed production of the candidate species is captivity is the only solution to make available quality seed in required time and number. Again, dependence on natural source for breeders for breeding purpose has also been provided impractical because of their non-availability. Even if you could be procured, many times they may not survive due to the injuries they have suffered during their capture. The broodstock fishes that are maintained, especially in ponds and tanks, do not breed by themselves in captivity. They are made to they are made to breed by themselves. They are made to breed in captivity by reproduction hormone manipulation, and this is called induced breeding. Induced breeding. The fertilized eggs that are obtained by induced breeding are incubated for the hatchling, and seed production is achieved by rearing them to the stockable size in culture ponds. The success in induced breeding and seed production of Indian major crops can be attributed to the fact that suitable technology have been developed for the development of their broodstock and induced breeding, whereas in the field of brackish water, finfish breeding and seed production, the technology have not been developed. Of late, the Central Institute of Brackish-water Aquaculture at Chennai has taken up the problem of broodstock development and breeding of brackish-water finfishes and has reported success in case of Miguelcephalus sea bass or and Lattice calcarifer and Eutroplus ceratensis. Other candidate species that can be taken up for the broodstock development and breeding are chonos, gropers, epinephrus, and so these are the some important candidate species of uh, breeding techniques. And coming to the next one is broodstock maintenance and development. The first step in broodstock development is procurement of sub-adult or adult specimens from the wild. So development of a broodstock from hatchery produced young ones will be time consuming and unnecessary. So usually subadult and adult both males and females are collected from commercial catches at landing centers. So prior arrangement have to be made with the boatmen or fishermen to bring them specimens in live conditions without any major injuries. Once the specimens are brought to the shore, uh, they are transferred to suitable containers having seawater. The container should be provided with aeration using portable aerators. If the time required to reach the destination, that is hatchery or farm, is more than 1 hour, it is advisable to transport them under sedation. On reaching the destination, they should be treated with suitable antibiotics for healing of minor injury, if any, and also for protection from any infection. The specimens are then acclimatized for 1 or 2 days at a later released into the broodstock holding facility after recording weight, length, and sex. In some fishes, the sex determination may be difficult, especially when the gonads are an immature and maturing stages. And coming to the next one is broodstock holding. So broodstock can be developed in water-based system or land-based system. In that first one, water-based system discuss like pens. So pen could be successfully used in protected bays or in estuarine lagoons for the development of broodstock. The pens either square or rectangle or semicircular in shape and 100 to 200 meter square in surface area can be constructed. Materials for the pen constructed depends on the local availability, weather conditions, susceptibility to natural uh, calamities, water currents, and bottom contour. The message of the nylon netting used for the pen construction can be of suitable message according to the size of the broodstock specimens that are going to be maintained in the pen. And after cages, floating cages are usually used for broodstock development. Cages made of polyethylene netting materials are attached to uh, uh, cage pipe or wooden frames kept afloat by uh, uh, siphonum drum and anchored within a calm bay or a sheltered marine environment. The size of the cages varies from 10 to 100 square meters. So, in surface area with a depth of 2 meters, like small cages are more suitable because they are easy to maintain and manage. The message of broodstock cages varies from 4 to 8 centimeters. The stocking density of fish is 1 kg per cubic meter of water. And another one is a land-based system like ponds and concrete tanks. Earth ponds or concrete tanks built on a land are also good for broodfish development compared to net cages. The size of concrete tanks used for holding broodstock depends on the size of the hatchery. So it is advisable to use a bigger tank to allow the fish ample space for swimming. So generally tank volume ranges from 100 to 200 tons, like 5 into 5 into 2 m, like 2 meters, and 10 into 10 into 2 meters. Stocking rate in a broodstock tank is 1 fish per every 2 cubic meters of water. Good water quality in broodstock tank should be maintained. Daily water exchange about 75% is recommended. And after feeds and feeding. Feeds and feeding have to be decided on the feeding habit and feeding habits of the broodstock. In case of mullets, like example, chonos, which are not predator, formulated pelleted maturation feeds are supplied. In case of carnivore species, which do not accept pelleted artificial feeds, or normally feed with low cost trash fishes. These trash fishes can be supplied in live condition and after freezing them in deep freezer. It may take few days to start accepting frozen trash fishes since live trash fishes can carry parasites to the broodstock tanks. It is good to feed in the frozen trash fishes both in case of pelleted artificial feed and trash fishes. Feeding can be done 3 to 5% of body weight of the broodstock. Feeding is normally done in two installments, that is in the morning and afternoon. So nutrition has a profound effect on gonadal growth and fecundity of broodfish. Although precise information on the nutritional requirement of gonadal maturation of various species is not well known, it has been recognized that quality and quantity of the diet significantly affect broodstock reproductive success, spanning, hatching, ability of eggs, and survival rate of the offsprings. So that is the feed and feeding of broodstock. And coming to the next one is major units of shrimp hatchery. So that is maturation unit. The maturation tank used in hatchery is generally of 10 to 16 tons capacity. 10 to 16 tons capacity. Round or rectangular with oval shape inside. A 50 diameter PVC. So that's mean polyvinyl chlorinated stand pipe regulates the water flow during 200 percentage of water exchange. The maturation unit should be soundproof and a photo period of 12 hours light, 12 hours dark is maintained in maturation unit. So diffused illumination for 12 hours a day is provided by fluorescent light of 10 mm, volts, each suspended and 1 meter above the tank. The total lighting stimulates like a deep sea environmental for breeding. And another one is larval rearing unit. The larval rearing section is one of the main area of activity. So the tanks used are of parabolic shape and are constructed with cement. The cylindro-conical or round flat bottom are made of uh, FRP material like fiber reinforced plastic material. RCC tanks may be also used. Capacity of tanks different from 2 to 10 tons water. Each tank has one outlet with a maximum slope drawn towards outlet which enable to drain out even the last drop of bottom water. So aeration from air grid is provided in each tank and throughout water supply uh, unit treated seawater is filled in larval rearing tank like LRT tank. That's mean larval rearing tanks. The inner surface of the tank is painted with green epoxy color. And another one is post larval rearing unit. The hatcheries having indoor post larval rearing facility use cemented tanks either of rectangular, flat bottom or a parabolic U shape of which the latter is more efficient. The capacity varies from 10 to 30 tons of water. The inner surface of the tank is coated with good epoxy resin painting of fiber coating for maintaining better hygienic condition. The post-larval unit is also well connected with water and aeration system. And after algal culture unit, algal culture unit. So algal section has got two wings like indoor algal section and outdoor algal section. Indoor section is completely air conditioned. As the space of indoor air conditioned algal unit is limited, so 20 liters to 200 liters carboli are shifted to outdoor algal culture section. For mass culture, 1 to 6 tons cemented tank or 200 liters FRP tanks may also be used. And after Artemia hatching unit. For hatching of Artemia cyst, 200 to 500 liters capacity. So FRP cylindro-conical shaped tank having bottom drain facility with a side glass window is needed. So continuous aeration and 40 volts light fixed at 20 centimeters 1 meter above tanker provided. And after counting and packing unit. So for counting and packing and transportation of seeds, separate section is stretched with the work. So nowadays it is just an open shed. A totally indoor rearing facility of the hatchery will need it. So 50 to 70 square meters area per million build up space. In a hatchery, shed protected by walls and roofed with asbestos sheet, asbestos, asbestos sheet, uh, interspersed with transparent FRP sheet for lighting. So this is the uh, briefing of total hatchery and hatchery different types of tanks and hatchery maintenance and brood stocks and brood stock maintenance. Is there any doubt?
No, sir.
Are you understand?
Yes, sir. Understood. Understood.
Okay. Okay. And another one is harvesting, seed packing, and transportation of seed. Like harvesting of larva or post larvae should be done carefully so as to avoid any stress to them. So harvesting is done by draining the water through the filter nets placed in the large containers so that water is retained in the net. The larvae collected in the net are removed periodically so to other containers so as to avoid overcrowding. Seed packing and transportation. Proper packing is very necessary for transportation of seeds. So polythene bags of 15 to 20 liters capacity are used for packing of prawn seed. So the corners of the plastic bags are rounded off with the help of rubber bands so as to prevent the prawn seed from getting trapped during packing. So for filling the plastic bags, the water from the water from the post larval tank is used. The use of new water is avoided for packing. If new water is uh, used, most of the post larvae will molt during transportation with consequent losses through the cannibalism. In order to provide shelter to post larvae, a few 10 plastic straw or nylon thread should be placed in the bags. To avoid cannibalism of post larvae during the long distance transportation, post larvae are fed with live feed such as Napoli of Artemia. So before putting them inside the bag, after feeding them properly, so the post larvae are transported in appropriate density like 75 to 125 per liter, depending on the distance of transportation. After placing post larvae, the hair present in the bag is removed by folding it, and remaining two-third of the bag is filled with oxygen by inserting the oxygen supply pipe in the water from oxygen cylinder. This helps the saturation of dissolved oxygen in the water. The bag is then sealed tightly with a rubber band. Transportation and after transportation of the post larvae are transported to short distance for duration of 4 to 5 hours in an open container without oxygen. So, however, for long distance of transportation of seeds, like up to 36 hours of journey, So oxygenated plastic bags are used. And transportation of shrimp post larvae. Transportation of shrimp post larvae is generally done in polythene bags under oxygen. The number of fry placed in the each bag will depend on the size of the larvae. So distance and duration of transportation. At PL, that means post larvae 15 to post larvae 20, about 2,500 to 5,000 post larvae can be placed in a bag with about 4 to 5 liters of filtered seawater. And lower density should be usually if fry are transported a later stage. During extended transport period, like water temperature must be lowered and packed the thermocol packing to reduce the rate of metabolism of the larvae. So this is the total uh, in hatchery management and feeding and transportation of post larval stages. Hello.
Yes, sir.
Okay, Ramay. Is there any doubts?
No, sir, no doubt.
Okay, okay. So remaining topic is continue to next class tomorrow.
Okay, sir.
Okay, Ram. Thank you. Only
Thank you,
one
sir.
candidate. Okay, okay. Thank you, ma'am.